Hello friends, welcome back to my channel Plant Pathology Pathsala. So in this video we will discuss about the late blight of potato. First about a brief introduction about the late blight of potato, then we will discuss in detail about the symptom, then its life cycle, etiology, then ultimately the management practices. So first, the causal organism of the late blight of potato is the Phytophthora infestans. It was first discovered by its life cycle was discovered by Anton de Berry. Remember this. This is the first pathogen, okay, on which this means life cycle study was done, and this Anton de Berry, who was known as the father of modern plant pathology, he discovered the life cycle of this Phytophthora infestans. Then this is not a true fungi. This is Oomycete fungi. So I think you know about the Oomycete fungi that it lack chitin cell wall. So the Oomycetes contain cellulose or beta glucan in the cell wall. There is a key differentiation between the Oomycetes and this true fungi, where the true fungi contain chitin in the cell wall. Other differences are also there. We will discuss this in the fungal classification part. Okay. Then major host for the late blight or this Phytophthora infestans is the potato and tomato. So late blight may occur in tomato as well as potato. Symptoms are similar only. Then this is a hemibiotropic pathogen. Hemibiotropic means, I have already discussed in wheat blast and rice blast that hemibiotrop means it acts as both biotroph as well as necrotroph. So initial phase of its infection, it behaves as biotroph. Means biotrophs are the pathogen that need a living host. They derive its nutrition from living host only, whereas the necrotroph, they first kill the host by using toxins or some other metabolites. Then after killing the host, they will derive its nutrition from the dead host or dead cells. So the hemibiotrophs are in between the biotroph and the necrotroph. So initially the pathogen behave as biotroph. They do not kill the infected cell. They just derive its nutrition from the living cell only, and then after some time it kill the cells and act as necrotroph. It derives nutrition from the dead tissue. Okay, so Phytophthora, Magnaporthiorizi, these are hemibiotropic pathogen. Then it is polycyclic. Polycyclic means in a single crop season, it can produce multiple disease cycle. Multiple number of means it's multiple number of generation it can produce. Okay. Then major plant part affected are the leaves, petioles, stem and tube. We will discuss in detail what is the characteristic symptom of this late blight of potato. Then historical importance, it is a major cause of Irish potato famine 1845. Very important. The causal organism or main cause of Irish potato famine is the Phytophthora infestans. And remember the year also. In which year this famine occurred? This 1845. It is one of the most destructive potato disease worldwide. And then under favorable condition, epidemic develop, and it can be extremely rapid. Okay, so this example is the Irish potato famine. Then Indian importance, major disease in cool humid potato growing area, mostly the hilly regions. Okay, because in hill region you will get the cool and humid climate. Then recent Indian studies reported that this clonal lineage EU13A2 is an important dominant clonal lineage for Indian region. Okay, we will discuss in detail about this. Okay, why it is important. Because you might have heard that one very important fungicide is there for Oomycete fungi, that is Metalaxyl. But nowadays the Metalaxyl is showing means the fungi Oomycetes, the Phytophthora, these are showing resistance to this Metalaxyl. This particular lineage, clonal lineage EU13A2, which is very dominantly found in Indian continent, are showing resistance to this fungicide Metalaxyl. Okay, so let's discuss this disease in detail. First, about the symptom. So we have already discussed that the symptom of leaf blight or late blight of potato was noted in leaf, petiole and tuber. So let's see what are the major symptoms. So on the leaf, initially you will get water soaked lesion, pale water soaked lesion you will get. Then that water soaked lesion later become enlarged and become necrotic. It will become brown to blackish in color, like you can see in this photo. And when high humid condition is there, on the downside of that necrotic lesion you will get cottony growth, white cottony growth you can see. This is nothing but the sporangiophore, which is containing the sporangia. This is the sporulation or sporulating structure of the fungi that look like white cottony growth, just similar as the downy mildew fungi. Okay, so this is about then this several lesions will coalesce and ultimately and it will cover the entire leaf surface and causing blightening, and ultimately plant will die. See, this is the major difference between blight and spot is, if this necrotic spot is restricted to a particular place, then we will just simply call it as leaf spot. But if many spots will coalesce and ultimately affecting a larger area causing heavy destruction, that will be called as blight. Okay, so this is the basic difference between leaf blight and leaf spot. If this spots will not coalesce, then we might have called it as late spot of potato, not late blight. Okay, then about the petiole and stem symptom. So elongated brown to black lesions develop on petioles and stem. You can see clearly here, brown to black lesions, elongated lesions are there on the petiole and stem, which later girdle the stem and ultimately the plant will collapse. Then on the tuber, so the sporangia or zoospore infect the foliage, reaches the tuber through the rainwater. And which tubers are infected? Where the earthing up is not done. Means earthing up is that when tuber formation start, we will put the soil to hide that tuber so that it will not exposed. Okay, so those tuber which are exposed, that are come in contact with the zoospore or sporangia from the infected foliage, because the first infection start from the foliage part or the leaf part. Here you will get this sporulation. So this sporulation are washed by rainwater and ultimately it will reach the exposed tubers, and on tuber irregular shallow reddish brown to black lesions are formed. Okay, here you can see reddish brown lesions are there that are irregular and shallow, and internal tissue so characteristic this rusty brown granular diffuse dry rot. It is not causing any soft rot, it is granular diffuse dry rot, rusty brown granular diffuse dry rot is there. Okay, and secondary organism may cause the soft rot, but this organism not causing this soft rot. Okay, so this is about the symptoms. So on leaf blightening will occur due to coalesce of this necrotic lesions, and on the downside you will get the white sporulation. Then elongated brown to black lesions are there on the petiole or stem, which ultimately girdle the stem. On tuber you will get reddish brown lesions, irregular lesions, and internal tissue shows the rusty brown, granular or diffuse dry rotting. Next, about the pathogen. So as I have already told, this is not a true fungi. This is Oomycete fungi. Mycelium is cynocytic or aseptate, no septa is there. Then sporangiophore is sympodially branched like this. Okay, alternate branching is there, sympodial branching. Then the sporangia, very typical, it is producing lemon shaped sporangia with a prominent papilla. Here this is a papilla. Okay, so the sporangia of Phytophthora is lemon shaped and with a prominent papilla. Then sporangial germination is affected by the moisture. So if moisture as well as the temperature. Okay, so if there is warm temperature, moisture is not that much there, then the sporangia directly form germ tube. But if that is the direct germination, but if it is exposed to, or if the condition is very favorable, cooler climate is there, high humidity is there, then it will germinate indirectly and form biflagellated zoospore. Okay, biflagellated zoospores are produced. The typical zoospores are actually kidney shaped, and this two types of flagella are there. One is tinsel type and one is whiplash type. Remember this. So this pathogen Phytophthora infestans produce biflagellated zoospore that is of kidney shaped. One tinsel and another is whiplash. Whiplash is posterior and this tinsel is the anterior one. Then two mating types are observed. It is a heterothallic one. Okay, A1 and A2. So if both these mating types are present, then only sexual reproduction will occur and it will produce the oospore. So two types of spore it produce. This sporangia or sporangiospore, that is the asexual spore, and the oospore, that is the sexual spore. Okay, so this is about the pathogen. Then about the disease cycle. So the primary source of inoculum is either the seed tuber, infected seed tuber, or infected crop debris where this oospores are present. Then it causes the primary infection, leaf spots or leaf lesions. Then on the downside, leaf sporulation, the sporulation will produce, the sporangia are produced, which are washed and disseminated by rain and wind, causing the secondary infection. So this is about the disease cycle. This is polycyclic in nature. It is having very short latent period. Latent period means the gap or interval between the infection to the sporulation. Okay, so if the latent period is short, means it is producing very abundant number of conidia or sporangia or spores within a very short period of time. So abundant sporulation is there and repeated infection cycle, that is leading to rapid epidemic development. Next, about the epidemiology. So the favorable condition for this disease is cooler temperature, 10 to 20 degrees Celsius. It requires very high humidity, more than 90%, cloudy weather, rain, prolonged leaf wetness, then dense crop canopy. Then why this disease is very destructive? Most important point is it is polycyclic in nature, because multiple infection cycles can occur in a single season, that is leading to heavy yield loss. Then short latent period, abundant production of sporangia, wind and rain dissemination, repeated secondary infection cycles are there, high genetic variability, it can overcome the host resistance gene and can develop fungicide resistance. These are the reasons, that's why this late blight is very much destructive. Then Indian status. It is this disease is very important for North Indian plain, hills and humid potato growing regions. Okay, and recent studies shows that this particular lineage EU13E2 is very important or dominant clonal lineage in India. This lineage is Metalaxyl resistant. I have already told this Metalaxyl is a systemic fungicide that is mostly used for the Oomycete fungi, but this particular lineage is showing resistance. Okay, so asymptomatic infected seed tuber can serve as important source of disease spread. Then about the global status, it occurs worldwide wherever potato are grown. Then the global movement of this pathogen lineage has changed the epidemiology. Major concern is the aggressiveness, fungicide resistance and breakdown of host resistance. See, this is very aggressive when the favorable condition is there, and most important thing is the adaptability of the pathogen. The pathogen is adapting to the fungicide. It is showing fungicide resistance. That is a major concern. And then breakdown of resistance. See, due to worldwide distribution of the potato, it is all the pathogen is also becoming worldwide distributed. Means its spread is very high, and ultimately, what happened due to the sexual reproduction, it undergo variability and new pathotypes are forming, new races are forming, that is leading to the breakdown of host resistance. Late blight remain a major threat to global potato production and food security. Next, about the management. As we all know that the primary source of inoculum for this late blight of potato is the infected seed and crop debris. So the major thing we have to do is we have to properly do the cultural practices so to avoid or to destroy the infected crop residue. Then we should use certified disease free seed, destroy infected tuber and cull piles, ensure good drainage because high humidity will favor the disease, proper earthing up should be there because exposed tuber will be ultimately got infected. Then about the irrigation, we should avoid the excess irrigation to reduce the prolonged leaf wetness because leaf wetness ultimately contribute to disease. Then the most important thing is the host resistance. Grow late blight resistant cultivars like Kufri Chipshona 4, Kufri Fryom, Kufri Currant. These are the resistant sources. Okay, you can use these varieties. Then chemical management. As I have already told, Metalaxyl is nowadays showing resistance to Oomycetes. So we should focus on preventive spray. So proper surveillance or forecasting of disease should be there so that we can do the preventive fungicide spray of mancozeb or chlorothalonil. And if infection is there, then we can go for the systemic or translaminar options like cymoxanil, then dimethomorph, flupicolide, then propamocar. So these are new fungicides that can be used in place of the Metalaxyl. Okay, remember this is cymoxanil, dimethomorph, flupicolide, propamocar. Okay, these are new fungicide that can be used, and as we all know, this pathogen is undergoes variability. So we means it adapt the situation more likely. So rotate the fungicide with different FRAC mode so that it will delay the resistance development. Then forecasting advisory service. So to predict the favorable condition, that is for Indian condition, we use this Indo-BlightCast system to predict the favorable condition. Based on the risk, we can apply the preventive or protectant fungicide. So timely targeted spray can be done, and it will reduce the number of spray, and ultimately it will reduce the cost of fungicide as well as the cost of cultivation, and better resistance management can be achieved. So this is all about the this late blight. Now one liner points. So the causal organism, this is Phytophthora infestans is a fungus? No, it's not a fungus, it's a Oomycete. Then what are A1 and A2? These are two mating types, because this Phytophthora infestans is a heterothallic fungi. So these two mating types are essential for the sexual reproduction and to produce the oospore. Then why is this disease favored by cool and wet weather? Because the Sporangia production need cool and prolonged moisture, okay, for its germination, for its infection as well as for its spread. Then why Metalaxyl resistance is important? Because it reduces the efficacy of key Oomycete fungicide. Okay, so it necessitates the FRAC based fungicide rotation. Then late blight versus this. Then why this late blight is polycyclic? Because multiple infection cycle are there in a single season, so we will call it as a polycyclic pathogen. Then characteristic symptom on tuber is rusty brown internal lesion, internal dry rot is there, and on the top or surface of the tuber you will get brownish irregular lesions. Okay. Then EU13A2, this is a clonal most dominantly found in India, which is showing Metalaxyl resistance. The major source of primary inoculum for this disease is infected seed tuber and also this crop debris or plant debris. Then best management strategy for the late blight of potato is use of healthy seed, resistant varieties, sanitation, forecasting, plus timely fungicide application and rotation. Then about the difference between late blight and early blight. So the pathogen for late blight is Phytophthora infestans, which is Oomycete fungi. But for early blight, it is Alternaria solani, which is a true fungi. Then lesions, about the symptom on leaves. So on late blight, we have observed first initially the water soaked lesion that ultimately developed to brown necrotic lesion, and on the downside you will get the cottony growth. But here in the early blight, brown lesion with concentric rings. See, like concentric rings of lesions we will get in the early blight. This is also known as target board symptom. Okay, this is a typical symptom for the Alternaria. Then favorable condition. So this late blight is favored by cool and wet or cloudy weather, whereas this early blight is favored by warm and dry condition. Then epidemic development. For late blight, it is very rapid. Entire crop can be killed in few days, whereas the early blight is completely slower. So the major losses is due to the late blight, not due to the early blight. Then tuber symptom. On tuber, rusty brown granular or diffuse dry rot will be seen in late blight, whereas in early blight, sunken brown lesion usually less extensive. Okay, this is the key difference between late blight and early blight of potato. I think now all these things are very much clear to you people. Still, if you have any doubt, you can let me in the comment section. Thank you.